So welcome to the Northern New England CTR seminar series. My name is Katie Modal. I'll be one of your moderators today. I'm a faculty scientist at the MaineHealth Institute for Research, and I study neuroregulation of bone turnover. And just a few housekeeping notes before we get started. We'll have several speakers today and a Q&A session at the end. And so please feel free to put your questions in the chat, and my colleague Andrew Fritz will help to moderate that. Andrew, did you want to say hi?
Hello, my name is Andrew Fritz. I am a faculty scientist in the Stein Lab, studying epigenetics in breast cancer and the role of RUNX1 in breast cancer.
Great, thanks. So I'd like to introduce our speakers next. So we have Dr. Frances Carr, who is a professor in the Department of Pharmacology and also a member of the Cancer Center at the University of Vermont. She's a core lead for the NNE-CTR Translational Research Technologies core. We also have today Erin Brown, who's a faculty scientist at the MaineHealth Institute for Research and the director of the Molecular Phenotyping Core, flow cytometry, and viral vector cores here, and also co-core lead for the NNE-CTR Translational Research Technologies core. Dr. Brown is also joined today by Moa Person, who is a sales manager at SimCell, and she's here to discuss the calScreener technology that he's been utilizing. And then finally, we're also joined by Dr. Doug Totges. He's a professor in the Department of Pathology and Laboratory Medicine at UVM, and also serves as the director of the Microscopy Imaging Center and the newly formed Center for Biomedical Shared Resources at the Larner College of Medicine. So without further ado, welcome everyone, and thank you speakers for being here.
Great. Thank you very much. I appreciate the introduction. I'm just going to take a couple minutes of your time to give a little bit of an overview of the TRTC, the Translational Research Technologies core, in case people aren't familiar with the breadth of what's actually offered here. The TRTC we think of is actually the bridge between our research resources, the state-of-the-art programs and methods and technologies offered here both at UVM and at MaineHealth. The TRTC provides investigators with support for research based on genomics, proteomics, lipidomics, cell and tissue analyses, as well as mouse models for translational and preclinical studies. The coordinated infrastructure of the shared research resources, education and training, seminars, across our institutions and communities. So we offer a breadth of programs throughout. The essential features of the TRTC include, first and foremost, I wanted to remind everyone, it's consultation with the directors and the scientific staff of the shared resource cores, whether it's at Maine or here at UVM. And importantly, when investigators want to explore their research and figure out what technologies, how best to think about the technologies that they may want to use in their experimental design, they meet with the directors and the scientific staff to explore whether that experimental design is going to be optimized for the research query that they actually have. And meeting with the directors and the scientific staff, you get introduced to a wide variety of methodologies and importantly, integrative approaches. So someone might come in thinking they just want to do a DNA sequencing, but in fact, they may be introduced to methods that they hadn't thought of before and approaches that are available in all of our resource cores in Maine and here in Vermont. So it's an educational opportunity as well. And importantly, the directors and the scientific staff provide an opportunity for assessment of the feasibility of the research proposal in the first place, with a specific attention to the rigor and reproducibility of results. And all of that's critical if anyone is going to be analyzing their data with the expectation for publication or for grant applications. Rigor and reproducibility is a high priority. And the shared resource cores also provide support for analyses, presentations, publications, grant applications. So it isn't simply a technology or a service center. It's a full service, if you will, full operation, soup to nuts to support our investigators. In addition to the research support, there is education, training, and outreach that's all of the scientific staff and directors are consider as a high priority. And such as seminars like this with introducing emerging technologies, applications, and novel research approaches. And it's important to share also that we have now got technology training, both virtual and in person. You can also send your samples to one of the cores and actually have an engagement with the conduct of the research in one of the cores virtually. So you don't necessarily have to drive across northern New England in order to share your resources, take access to the resources. And the core facilities actually provide an important opportunity for collaboration as well. And finally, we have to think about our shared resources really as innovation incubators to enhance network collaborations as well as access to state-of-the-art research methodologies. And one such innovation is actually happened here at UVM with consolidation of leading research core facilities into the Center for Biomedical Shared Resources, which happened just the last year and a half, 2 years. And the idea of bringing genomics, proteomics, flow cytometry, and microscopy together is to offer innovative, dynamic, integrated model of core facilities and opportunities to engage across the cores, bringing together these different approaches and developing innovative methodologies. So really, we're looking at this based on team science. The CBSR provides pioneering approaches and technologies to investigators and trainees. And it's important to remember that this is across the region. It's for all of our partners and members of the NNECTR. And with that, I'm going to turn this over so you can have an example and good discussion about some of the innovative technologies that are available through the TRTC. I will stop and turn this over to Erin.
And I will turn it over to Moa. I think she's going to go first, and then
you.
I'm going to go second.
Then I go first.
Yep.
Good transition with technologies. Can everyone see my screen now well?
Yeah.
Perfect. So my name is Moa. I'm, as I said, a sales manager at the Swedish medtech company Simsol, and we are offering a fairly unique technology called the calScreener that I will tell you a little bit more about during this talk today. So fundamentally, all cells and all biological processes, as you know, they are metabolically active and they produce heat. And our technology can measure this heat production with extremely good sensitivity. And actually, this is the only method that can directly assess direct heat production. And the heat per se is directly proportional to the metabolic activity, which is being measured in real time. So why is it relevant to measure overall metabolic activity? Actually, we have up to date presented in more than 30 publications that this can really reveal a lot of important insights that is not visible by solely traditional methods. So it provides a lot of important insights. And the instrument, it looks like the picture is being showed here. It consists of a 48 well plate format where we have developed these unique cell culture and microscope ready inserts to allow a better cultivation condition for your cells. And this is all combined with a quantitative and qualitative software to allow intuitive data interpretation. One of the biggest advantages the instrument brings is that it actually allows the possibility to measure literally anything, any type of media and any type of compound. And basically anything that fits into one of the vials we can measure, and we do that in real time. So it really allows the possibility to, in a fairly high throughput way, screen different conditions and different samples. The method is completely non-destructive. So it also opens up the opportunity to run post-experimental analysis directly, which is, of course, a very important aspect for many type of preclinical studies. The first theoretical example I wanted to show you from the instrument is this, where E. coli has been cultivated with a traditional CFU counting approach, which you can see in the dotted line. In parallel, it has been measured in one of the calScreener vials. Interestingly enough, you can here see that the maximum metabolic activity is actually in the stationary phase, where there's no changes obtained in biomass. So the reason why I'm showing this is because it provides a really nice proof of concept that we can reveal a lot of important insights that is not visible by traditional or conventional methods. The following example is, you can see first here to the left, it's 4 different strains under the same cultivation condition. And what's very interesting here is that you can see that each and every one of these strains, they give rise to this very unique metabolic fingerprint. This is something we have confirmed is extremely reproducible and is something that has brought a lot of value, particularly in the field of infectious disease. To the right here, you can see instead E. coli, but under different cultivation conditions. So this is very much showing that the instrument is also sensitive enough to pick up these differences under different cultivation conditions, which can also bring values in many different aspects in terms of answering a lot of important questions. I will now start with sharing a couple of examples relating to microbiology, AST testing, and infection disease. The first example I wanted to share is a publication where our instrument was evaluated as a more effective way of evaluating the efficacy of combination treatment. And obviously combination treatment is a very important target to address MDR isolates, but it's extremely time consuming to evaluate with conventional methods. In this study, hundreds of different MDR isolates were analyzed together with different combination treatment. And that was being done with the calScreener, conventional MIC methods with CFU countings, as well as a comparison in a marine infectious model. Foremost, it was confirmed in the study that it was a very good correlation between the metabolic values obtained in the calScreener with traditional MIC methods, which very much confirmed that the calScreener actually can be used with the purpose of finding a more effective way of evaluating these type of combination treatment. What was also very interesting was that there was a direct correlation between the thick metabolic index values and the CFU counts in peritoneal fluid in the marine mouse model. And this was a trend that was not actually obtained when looking on traditional CFU countings. So the reasoning in this article was that the calScreener foremost can provide a more effective evaluation of antibiotic combination, but also providing a better in vivo predictability. The following example is from a professor called Tom Connier, which is very leading in Europe within the field of biofilm and chronic infection. And one of the main reasoning here is that one reason that gives rise to resistance within biofilm is a reduction in metabolic activity within the biofilm, which is obviously something that cannot really be addressed with traditional methods. So in this study, Professor Connier was evaluating different carbon sources as possible potentiators to boost the effect of different antibiotic. And in the example here in the graph is on sodium acetate. And first of all here, biofilm has been formed directly in one of the calScreener vials and grown in the vials. After that, sodium acetate in this case has been added together with combination of antibiotic. And here it can be quite interestingly see that sodium acetate seemed to have a boosting effect of the metabolism here, which was an effect that exactly what he wanted to investigate. This was also compared with traditional CFU counts where a reduction in CFU actually also could be seen. And in overall, this really shows that the calScreener can help reveal these insights and actually evaluate these type of potentiator in a way that other methods cannot do. Another important thing that Professor Connier highlights is that the calScreener brings the capacity to actually measure biofilm in its fully intact form, as it's shown here. It can do so in any type of media. He is particularly interested in using cystic fibrosis media. and it is real-time measurement. So it overall provides a very effective way of evaluating biofilm and chronic infections. The following example is from a researcher at Karolinska Institute here in Stockholm, Sweden, that has a particular interest in looking into different type of complex model. So here you can see an organotypic skin model, so a co-cultured model that has once again been grown directly and placed directly in one of the calScreener vials. And it has been infected by Streptococcus. They did a follow-up publication where they were interested in looking into different combination treatment in order to evaluate the treatment efficacy. And some of the most important findings in this article was that the calScreener provided a much more quantitative readout that could reduce the experimental variation that was obtained by using CFU countings. And the group also very much emphasized that the calScreener brings a very interesting way to study these type of models without disturbing nature's true architecture, but really maintaining the natural form of this organotypic models. I will now share a couple of examples within the field of cancer biology. And one of the researchers that we are working very, very closely with in this field is Dr. Stephanie Gross from University Hospital of Basel. And in this example, she's very much emphasizing that there is a big challenge of finding good preclinical models that allows for rapid screening of drug efficacy in cancer inhibitor, particularly directly in patient material. So she was using the calScreener for this purpose in this article, and she could confirm foremost that the calScreener provides a sensitive way of actually measuring treatment efficacy, and foremost directly in patient materials. So she was adding fully intact tumor slices from patient and evaluating the treatment efficacy. So with these findings, she really emphasized that the calScreener brings a very interesting approach to study and allowing more individualized, rapid drug response assessment that really plays a crucial role for finding more personalized medicine. Another publication also done by Dr. Gross, but with a similar focus, but where she used the calScreener to evaluate the hypothesis that cancer cells has an increased and a higher thermogenetic capacity. This is once again, of course, also something that's very tricky to investigate with traditional methods. But as our instrument actually allows the capacity to directly measure overall thermogenesis, she was in this article comparing primary cell lines with metastatic cell lines, and where she, interestingly enough, could see that the metastatic cell lines actually did have a higher thermogenetic capacity. So it was the first time she were able to address this question directly, and she highlighted that the calScreener with this information really provides, once again, a powerful tool to understand the metastatic potential, which is an important part for more personalized medicine within this field. So to briefly summarize, our instrument provides a very unique capacity to measure overall heat, overall metabolic activity in real time, in any type of sample, and in any type of condition. And with these examples I've showed you today, we have really seen that this can enhance preclinical research in many aspects and help pave the way for more targeted therapies. And now I wanted to hand over to Aron for him to share a little bit more around what he have been using our instrument for in terms of adipocyte tissue research.
There we go. Okay. So yeah, I'm just going to tell a quick story about how my lab has been using the calScreener to measure energy expenditure in adipose tissue. I'm particularly interested in brown and beige adipocytes, as opposed to most the type of fat you usually think about, like belly fat. Brown and beige are unique. Instead of storing fat, they can actually burn fat and release that fat as energy. And brown and beige fat is typically activated during cold exposure. And so it maintains euthermia and prevents hypothermia. And so you have both mice and humans have white and brown fat and beige fat. Brown fat is typically a permanent tissue that is always on, whereas the beige fat is a subcutaneous type of fat. So here in the mouse here, this inguinal fat would be subcutaneous, and that can actually turn to a brown-like fat during cold exposure. And what happens, why this fat is unique, is it actually expresses a specific protein called UCP1, and that uncouples oxidative phosphorylation such that ATP is not made. And these protons leak across the mitochondrial membrane, and that energy has to go somewhere. That gets released as heat. So when your brown or beige fat gets activated, basically what you're doing is you're warming up your blood. And so what we're trying to do is figure out ways to keep these cells active without the cold. And that could possibly be a way for causing weight loss in patients with obesity and type 2 diabetes. And so we've developed this hypothesis in my lab where we think in an unactivated beige adipocyte, there's microRNAs that normally block thermogenesis. However, when these cells are activated either by cold or by a drug, these microRNAs, their expression goes down really quick within less than an hour. And we think that these are being secreted by exosomes. And so we think this exosome pathway could be important for beige adipocyte activation. And so we turned to a mouse called the RAB27 deficient mice, and to study if there's a role between exosome secretion and thermogenesis. And exosomes are formed in microvesicular bodies. And what RAB27 does, it's responsible for fusing that microvesicular body to the cytoplasmic membrane such that the exosomes are released. And so we've taken beige adipocytes from these mice and cultured them and then, taken the conditioned media, which contains these exosomes, just to see if we can see a difference between the mutant and the wild type mice. We measure them on this particle analyzer called the ZetaView. And you can clearly see that on the left here is the particle counts. These are the exosome counts. And on the bottom of the graph, this is the size of the exosome. But clearly the wild type beige adipocytes are producing about 10 to 12-fold more exosomes than the het and the null mice, beige adipocytes from those mice. When we look at these beige adipocytes in culture and we treat them with a drug called forskolin, which activates the cells, it's sort of like simulating cold. UCP1 expression goes up in the wild type, and you can see that is blunted in the het and the null mice. And so we want to find out, is this decrease in UCP1 actually causing these cells to give off less heat? So now we've turned to the calScreener to answer that question. So we grow the beige adipocytes from these mice in these little calScreener inserts. And if you look at the heat profile, so this is accumulated heat over time, over a period of about 17 hours. You can see that the wild type mice are giving off quite a bit more heat than both the het and the null. And that can be summarized over here by total heat. The other thing that we've been really interested in is, besides just growing cells in culture, can we actually harvest tissues from these mice and detect differences in heat production? And so one of my colleagues, Matt Lyons, harvested some fat tissue for us. He took interscapular brown, the inguinal, which can give rise to subcutaneous fat that can give rise to beige adipocytes, and the gonadal. And generally the brown will always be on at a certain level. Some of the beige will be on because even at room temperature, some of the subcutaneous fat will brown and turn into beige. And then the gonadal adipose, it's more like your visceral fat, generally is pretty refractory to any sort of thermogenesis. And so we just put pieces of these fat tissues in the calScreener and ran them. And you can see that the brown, it has a much higher metabolic rate. It's giving off much more heat, followed by the beige or the subcutaneous, followed by the visceral. So we think that the calScreener can be used to distinguish different fat tissues based upon heat production. So we've turned back to our RAB27 mouse model. And we've basically taken these mice, RAB27 wild type and het mice. We're actually working on the nulls right now, but we put them in our metabolic cages that we have in our physiology core. And these cages can actually measure the energy expenditure in these mice. And so you can see both the wild type and the het mice at room temperature have a similar amount of energy expenditure. When you put them in the cold, they both respond, they both burn more energy, they're burning more calories. But there is a slight significant difference in energy expenditure in the het compared to the wild type. And if we resect these adipose tissues, these are the inguinal ones, and we actually do qPCR on these, just like I showed in the cell culture, you can see that upon cold stimulation, this inguinal adipose tissue is expressing much more UCP1 compared to the het and the null. And so what we've done is we've sectioned these pieces up into smaller pieces of fat to put in the calScreener. And you can see over here on the right that there's not much difference between the wild type and the het, but you can see that the wild type is responding pretty good. So these are just mice that were in the cold for 7 days. We cut the fat out, put them in the calScreener, and the wild types are basically producing about 20% more fat, more heat production than those from the heterozygous mice. So just to summarize, I think the calScreener can be used to measure heat production in cultured adipocytes. The heat production in these cells correlates very well with the UCP1 expression. We can measure heat production in live resected adipose tissues. I'll mention a few drawbacks, and one of them is it does take a while to seal the lids. So while you're putting your samples in, you know, you're going to lose some heat. And then when you first put the cal plate into the machine, it has to equilibrate and it has to warm up to 37 degrees before you get, there's a lot of frictional heat when you push it in. You can see that here on this graph. And then you get to this steady state, and that's where you start measuring. So that takes about a half hour. So you lose 30 minutes when you're putting your samples into the machine. And so I think it would be great if you could put the samples in the machine and then inject a drug into these little vials after the fact, because I think a lot of activity happens during that first 30 minutes. And I'm hoping that the company might sort of go down that route in the future. I currently have this instrument in my lab. I'm renting it. I have it here. I've had it here since last year, but I have it for about 3 or 4 more months. So if anybody's interested in using it, I'm happy to do any type of run, whether it's bacteria, tumors, fat samples, whatever. So, and hopefully one day we'll be able to purchase this instrument. That's all I have. Now I think we're over to Doug.
Are you doing questions first, Aaron, on your presentation? No, okay, Fran says no. Okay. Okay, so thanks everyone for being here. I assume you can see my screen. Thumbs up. Yes, thank you. Okay, so yeah, thanks for the invitation to do this. This is I think an exciting time for us here at UVM and working together with our colleagues in Maine. And I thank Fran for the introduction on the Center for Biomedical Shared Resources. And this, I think the introduction of the center came at a really opportune time for us, because the technologies I'm going to speak a little bit about today are in the realm of spatial omics. And spatial omics is one of the newer omics terms that has come to the forefront in science in the past few years. And we can really think of it as the ability to combine the molecular analysis of specific cellular analytes such as RNA and protein in the actual context of the tissue microenvironment. So I like to think of it, I usually frame everything around cycling in my own life and bicycling. And I like to think of it that, you know, on the left-hand side, we have the deconstructed parts of a bike, and as they're deconstructed, it doesn't really give you an awful lot of information about what a bicycle is. So we need to really compile them all back together and put them together in the final output of the bike. And I think that's the way I like to think about the spatial omics, is that we're going from individual analytes within a cell and trying to bring them back together into the whole concept of a tissue section. So within the CBSR, we have several methods that are within the realm of spatial omics. On the left-hand side here, confocal spectral imaging with tile scan and analysis, 10x Visium down here at the bottom. The two I want to focus on today are two of our newer technologies, and those are the Nanostring GeoMx and the PhenoCycler-Fusion, which used to be called the CODEX from Akoya Biosciences. So I'm going to start with the Nanostring. And the Nanostring GeoMx is referred to as a digital spatial profiler, a DSP. This is the instrument on the left-hand side shown here. Almost looks similar to the calScreener. Everything seems to be coming in a box nowadays in the labs, makes things easy to implement and to store. But the way in which the instrument works is shown here on the right-hand side. And basically, it's a methodology that utilizes oligonucleotide tagged probes and a fluorescent marker together with them. So essentially, it's very simply staining slides, defining regions of interest. It then uses a UV radiation clip to remove the analytes. They get deposited from regions of interest into a 96 well plate, and then they're read out either on what Nanostring has an instrument called an N counter for looking at the probes, or you can do next gen sequencing, which is what we'll do in the CBSR. So the technology really is state of the art. This is technology that is being implemented in many different types of multiplex or high-plex spatial omics technologies today. And that's using oligo-labeled antibodies or oligo-labeled probes. So the GeoMx system allows you to look at either protein or RNA within your sample. If it's a protein-based sample that you're interested in, you'll have an antibody that has been pre-conjugated with a oligonucleotide barcode. It has this UV photocleavable link. You'll incubate your tissue section with this probe, and then the region of interest will allow you to apply a UV light that will cleave the linker and deposit the barcode into a deposit the analyte into a multi-well plate. If you're looking at RNA, it basically works the same way, but rather than using the oligo-labeled antibodies, it uses an oligo-labeled probe. Technology is basically the same. So to step through it, how this system would work, we have a high-plex mixture of reagents. So again, either protein, labeled antibodies, or labeled RNA probes. These will all be put into, mixed together in a tube. They will then be used to label a slide, all of the different probes on one slide section. And then regions of interest will be selected, and areas of illumination will be selected from that. UV light will cleave the probes off, which will then be gathered. And so then you can, as I mentioned, go on and do something like next-gen sequencing that's shown on the right-hand side here. The benefit, or one of the nice, I think, aspects of the GeoMx and the PhenoCycler that I'll talk about in just a few minutes is that you can do multiple different types of samples. Oftentimes, technologies are limited in that you can use a frozen section, but maybe not a paraffin section, or vice versa. In the samples that can be used for the GeoMx can be core needle biopsies, conventional FFPE tissue block, tissue microarrays, frozen tissue section, or a slide mounted tissue. So you really have the unique opportunity when you're planning an experiment to either acquire tissues fresh and then choose the type of preparation that you'd like to use, or you can go back into archived blocks and use those samples as well. The image on the bottom right-hand corner here is just showing the area on a slide that is actually used for the illumination of the sample. So again, multiple reagents are used in this system, and imaging reagents up to 4 colors. So what you want to be able to do is to locate your cells of interest within the tissue sample. And you do that by using up to 3 antibodies, and then a DNA marker. So you can choose the specific antibodies that would localize, for instance, epithelial cells in a carcinoma, or you could do immune type cells. That will give you then the target sample that you will then go in and cleave away the probes that have bound. So here's the overall workflow. So tissue slide is stained with the oligoconjugated antibodies or RNA probes. You then would, once they're all incubated together, you image the slide and select regions of interest based upon your specific antibody markers, and then UV cleave the oligos off of the antibodies or off of the RNA on the slide. Those probes are then aspirated away, and then they are collected into a 96-well plate for each region of interest. Once that occurs, then there's a handoff jump back there. This all occurs steps 1 through 6. So all of these steps are occurring or will take place in the microscopy imaging center in the CBSR. And then the step 7, which is the sequencing and counting, will take place in the Vermont Integrative Genomics resource, which is directed by Julie Dragon and actually is located right next door to us in the new CBSR. The tissue sections themselves can also be prepared by the staff in the microscopy imaging center, or they can be prepared in your own lab or cut in a hospital. But again, the beauty of this is that you could either, for folks, our colleagues in Maine or off of the UVM campus, can send or mail to us paraffin blocks or slides that have already been prepared. And so we can prepare those here and then do all of these steps. And then the data analysis is assisted then by the bioinformatics group, which is also directed by Julie Dragon. So the readout assay, as I mentioned, Nanostring has produced and sells something they call an N counter for doing the final assay readout. We won't be using that. We'll be using an NGS system that's located in the Integrative Genomics, again, right next door. So the plex capacity for this is greater than 20,000 RNA probes, and can do, for instance, RNA content. We can do the whole transcriptome or transcriptomes for cancer or for neurobiology at this point. Protein content can be 96 or more probes. And the entire process takes around 5 days. So this is a nice project work plan that Nanostring has put together. And this is the basis that we will use in the CBSR if an investigator contacts us about the potential of doing one of these projects. So we would have a form like this filled out with the study title, the rationale for what the study is about, specific aims, the application type, but then really study details are important. The species, so right now they have probes for both human and mouse. So the tissue or tumor type, format, what type of analyte you're looking at, protein, RNA, or a combination of the two, and then the custom content, which are the morphology markers. And these are a set of marker panels that Nanostring cells that allow you to do specific types of applications. So the target 1, 2, 3, 4 depends upon if you're looking at cancer cells, or if you're looking at, again, something like neuroscience. And then there's a region of interest, the way in which those are selected, and I'll talk about that in just a minute. So this would be the actual work plan strategy that we would design. This is in a little bit more detail. So when we design these experiments, and I must put in a proviso, is that we haven't done this yet. So the instrument was just installed about, well, a few weeks ago in our facility. The training on this instrument will take place actually next week, Tuesday through Thursday. So we haven't done an actual experiment yet. So this is kind of new for all of us. So we're spending a lot of time thinking about how you design these experiments, because the probes are expensive, and we want to make sure that we are understanding from the beginning of an experiment whether this is the correct approach or not. So this is just a little bit drilling down a bit deeper. If you came into us and set up a consultation, or if we did it remotely, the tissue type, what are you using for the type of sample? What is the species? Again, it's been GeoMx validated for human and mouse, number of slides, numbers of regions of interest, and areas of illumination that we would talk about. And it depends, again, upon the type of sample that you have. Again, we'd choose the species. You would choose, we're doing NGS readout, so the protein can be immuno-oncology or immunology markers or neuroscience markers. You're also allowed to do custom targets. So if you have specific targets in mind, they can customize. If it's an RNA experiment, there is immune pathways through the nCounter, but we'll do, again, the next gen sequencing readout. They have a cancer transcriptome atlas and a human whole transcript atlas and a mouse whole transcript atlas. So the human whole transcript atlas is about 18,000 probes. The cancer transcriptome atlas is around 1,800, so about 10 times less. Can also do custom targets. And then really important in the experiment is designing your regions of interest and your areas of illumination. So basically, there are multiple ways to do this, and I'll show that on the next slide. But your regions of interest are really detected by the morphology kits that we purchase from Nanostring. So all the kits contain a nuclear stain and 2 fluorescently labeled antibodies for our ROI selection. Again, you can add a third additional custom antibody for your specific application. The regions of interest can range in size and shape from about 10 to 600 microns in diameter. And as it mentions here, although regions of interest can be as small as 10 microns, about the size of a cell, the throughput isn't really conducive to single cell analysis because of the number of cells you actually need to do quantification of transcripts and proteins. So this is the profiling modality. So this is how region of selection would actually work. So the morphology reagents selected guide the region of interest selection. And they have, there are 5 different ways in which regions can be selected. Geometric, so this would allow you to assess heterogeneity by using different shapes that you create and then use to choose your region of interest. You can evaluate specific compartments by doing segmentation. So looking at one area, say the tumor versus the tumor microenvironment. Can look for rare cells, contours, or gridded. So there are multiple ways to choose the areas that you actually want to use off of a slide. And it's important to consider that before the actual experiment begins. Another way in which you can do your region of interest selection, and it's a little bit more clumsy, I think, but it might be an appropriate way to go if you don't have the best morphology markers, is to take a serial section from your sample and to do an H&E stain. And then if you create or acquire an image of the H&E stain in the serial section in the GeoMx, they have software that allows you to overlay them. So you could overlay and have a pathologist determine which are the correct areas then to choose for a region of interest to proceed. So once the once the sample has been stained and imaged and the probes have been cleaved off, then it goes to the NGS readout, and this is just showing the steps in that: the library prep, sequencing, data processing, and the counts. So the illuminated areas get put into specific wells in a 96 well plate, and those specific wells are then mapped back to the actual image to do the final SpatialOmics determination. And then the data analysis is, again, we would be working together with our colleagues in bioinformatics to do analysis, and there's software packages available for doing the final, rather complicated but detailed analysis. Okay, I'm going to speak then for a few minutes about the PhenoCycler. This is a second system, and this used to be referred to as the CODEX, which was an acronym basically for code detection by indexing. And the PhenoCycler is really a system for doing multiplex tissue imaging based on fluorescence and DNA conjugated antibodies, so somewhat similar to the GeoMx. It can capable of mapping about 1 million cells in 10 minutes, and it's the integration of the microfluidics shown on this side of the instrument together with a microscope, which is now actually a whole slide scanning slide imager to scan in the entire area of your section. So this looks a little bit busy, but really it's just showing the technology that the Akoya fusion cycle uses. This is from a paper in Nature Protocols from Black et al. And it's just showing how the probes are actually made. And you can make your own probes. You can conjugate with a DNA oligonucleotide on your antibody, and you can add the fluorescent probe, or you can purchase as well from Akoya. So basically the steps involved in this system are to iteratively hybridize, image, and then strip fluorescently labeled DNA probes that are complementary to the tissue-bound DNA conjugate antibodies. The limit on antibody markers is actually set today by the number of known unique DNA oligonucleotide sequences that do not exhibit tissue, cellular DNA, or oligonucleotide cross-reactivity. Today, it's around 60 available antibodies, but the panel is expanding as new oligonucleotide sequences become available. And it's typically, the belief is that there'll be more than 100 probes, unique probes available in the near future. So this system really works. It works with much like the Nanostring GeoMx, you can use FFPE or fresh frozen tissue samples. And basically the tissue samples are stained with an antibody panel that would be provided by Akoya, or that you can create your own customized antibody panel. Then a multi-cycle reaction takes place. So basically you hybridize with 3 probes plus a DNA probe, and then you image on the microscope. Those probes are then stripped away chemically, and then you rehybridize with a second round, image, and you can do this iteratively multiple times. And once the images are acquired, each step, image processing then allows you to merge the images together and do the SpatialOmics analysis. So results have shown that the iterative process, even though it's a chemical stripping, does not tend to denature the samples. It may depend on the type of sample, but multiple iterations up to multiple dozens have been published without showing any appreciative degradation of your tissue sample. So the tissue format is, it's a whole slide imager, so you can do whole slides, tissue microarrays, and tissue sections. There's the resolution. The throughput is just one slide at a time. Multiplexing up to 100 biomarkers, again, depending upon the barcode compatibility. And that talks about file format as part of the output. So the probes that you would acquire from Akoya are the CODEX antibodies. These are conjugated antibodies, again, for building antibody panels. The CODEX reporters are fluorophore conjugated oligonucleotides for visualizing the CODEX antibody that will bind. And then you can purchase your own CODEX barcodes, which you can conjugate to your own specific antibodies to do the staining. And again, this just shows the output and the steps required for doing the staining. This is an example of from Kara Lee in our in the Microscopy Imaging Center, and this is work she's doing together with Prachi Goul and with from Gary Stein's lab, Janet and Gary Stein. And this is just showing on the left-hand side here is a whole slide image of a section of human tonsil, and then it's stained through, in this case, it went through 2 iterations of staining and stripping. And so it has 6 probes on it because you do 3 antibodies at a time. So that's shown here together with the DNA marker DAPI, and it shows the probes that were used in this particular study. So CD8, CD20, CD107A, Ki67, pan-cytokeratin, and CD68. So you can localize these specific cell types within the tissue sample. This is showing up on the top here is a zoomed in image from the box shown on the whole slide image. You can see the various stains. The color on the image corresponds to the color of the key code here on the antibodies. And then once the staining has been accomplished and you have multiple images to analyze, we've been using Indica Labs Halo software, and that's shown down in the bottom right-hand corner. And this is showing an area from right around here on this section, which has been imported into Halo, and then the various cell types have been localized based upon antibody staining and nuclei to do a quantitative and a spatial analysis. And the Halo Labs allows you to do really in-depth, detailed analysis of the location of specific cell types, how close they are to one another, whether they cluster. It's incredibly complicated, or it's incredibly robust how many complicated types of analyses can be done. So again, the SpatialOmics options, as I mentioned, and the beauty of all of these different spatial options that we have is that we can perform all of the staining in an automated way using the LicaBond RMX auto staining, which is shown here. This is Kara. I took this picture today. She's got her fighting pose. Kara is also an accomplished MMA fighter, and so she has lots of images on the internet, and she has her pose. But nevertheless, this is the LicaBond, and the beauty again of that is the automation, which makes rigor and reproducibility much higher. As Fran mentioned in the introduction, we're all about rigor and reproducibility, and this system allows us to maintain that and to minimize potential technical errors. And so just going to finish by saying, if you have specific omics project questions, you can certainly contact me directly. We use, as Aaron's labs, cores use in Maine, we use a core organizing software called iLab, and this is just showing one of the tabs where you can do a service request, you can initiate a request, and then for a consultation. And our consultations, at least with the Nanostring GeoMx, include a Nanostring technical representative. And we've already done at least one of those, and it's very helpful because she, from the standpoint of Nanostring, can really hone in on specifically what is needed for the specific or for the application that we're planning to do, and tell us whether it really is feasible or not. So that is the consultation aspect for this. We will be doing, as I said, the Nanostring was just installed. The training next week, and sometime probably in April, or it may go into May, we'll be doing well, I guess a Nanostring festival, a day-long series, a day-long program here at UVM, where there will be representatives from Nanostring on site to hold in-person consultations. We could also do them remotely via Zoom for our colleagues in Maine, and to talk about specific projects and applications. So I think when that date is formalized, hopefully Sheila will be willing to send out to the NNECTR, thank you, Sheila, the NNECTR members when that's taking place. Okay, so that's what I have. I'm going to stop sharing. And I guess we have some time for questions. So I will let Kate, and I'm not sure who's going to be running the question and answering period. Oh, okay, sorry. Yep, sorry, okay.
That's fine. So if you have a question and you haven't put it in the chat, please raise your hand using the feature in the Zoom. One first question for Doug. RNA easily degrades. How do you maintain that in the tissue processing for the GeoMx?
So that, that's a good question. There are so many specific reagents that are used now to prevent RNA degradation, RNA away, and all kinds of all kinds of reagents that we use. But basically, during the processing for the GeoMx, we use depth seawater for all the reagents. It's just typically being careful of eliminating any type of RNases that may be present. And the results from the studies, I mean, I think that Nanostring has approximately 7,000 publications out there now on latest read. So Grace, I'll be happy to speak with you in more detail, but it's just optimizing your reagents and keeping things very clean.
Okay. Second question comes from Chris Franklin. Does the investigator provide the antibodies for use as markers for particular cell types? And I believe this was during the GeoMx part.
Yeah, thanks for the question, Chris. So as I showed on the GeoMx, they have several panels of markers that they sell for specific applications that one can use for the morphology markers, with the ability to add one of your own specific antibodies. If none of the panels correspond to your specific needs, they can provide conjugation of your antibodies with their probes, or you can do it yourself, or we can do it. So yes, that's certainly creating a panel for your own application. It is something that can be done.
Okay, there are no other questions. I'd like to thank everyone for joining us. Our next seminar will take place at noon on April 14th, and it is entitled Community Engagement and Outreach in the NNECTR, the what, why, and how. It will be presented by our core leader, Dr. Jan Carney, along with 3 of our rural navigators, Elizabeth Woods, Emma Daybranch, and Carrie Sullivan. Please plan to join us on April 14th. Thank you so much.
So I'll stay on. People can get off, but I see there's at least one more question from John Boyson. So yes, John, the answer is yes.
Okay.
Very simple. Sorry.
Oh, thanks. I was just looking it up on the website. I don't think it's on the website yet. Right?
It is not. It's
okay.
not. Yeah, but it can be used. It's not housed in the CBSR properly. Proper right now. It's in Dr. Stein's lab, but he has graciously opened it up for core usage. So
gotcha.
I think if you shake your head, then but
I'll verbally say yes. The other thing I was going to say is we've made a lot of our own conjugated antibodies. It's not very complex. No,
right.
We haven't. Kira and
Kira, Kira and Prachi.
All right. Thank you.
Okay.